Good afternoon, everyone. Welcome to the first Wednesday afternoon lecture of the academic year. We were so excited that we had to have it on Monday. And there will be, of course, during the course of the year, on Wednesdays at 3 o'clock in this space, a lineup of remarkable speakers. Today's remarkable speaker is indeed a pleasure for me to be able to introduce. Dr. Corey Bargmann. Her current position at the Rockefeller is as Torsten Wiesel Professor and head of the Lulu and Anthony Wang Laboratory of Neural Circuits and Behavior, and co-director of the Shelby White and Leon Levy Center for Mind, Brain, and Behavior. Corey began her training getting a BS in biochemistry at the University of Georgia, and then went on to a PhD at MIT with none other than Bob Weinberg, where she did some very interesting work on the gene called New. Deciding that maybe cancer wasn't the answer to everything, she then did a postdoctoral research program with Bob Horvitz. You notice she's chosen rather well here in terms of laboratories to work within, and became, as you will find out later in the course of this presentation, captivated by what one might be able to learn from the roundworm, C. elegans. She then spent some time, like 13 years, at the University of California, San Francisco. Along the way, became an investigator of HHMI, which she continues to be to this day, and then moved to Rockefeller in 2004. Her work has been just at the top of the field of understanding how neural systems work, and she has really been cleaning up here in the last couple of years, having been awarded the Kavli Prize in Neuroscience in 2012 and a Breakthrough Prize in Life Sciences 2013, which maybe someday she'll tell you about the experience of how she found out about it, both of which are extremely well deserved. I have to say, however, that I'm particularly grateful to Corey for the way in which she has put probably a chunk of her lab's efforts somewhat on hold, or at least a little bit pushed aside for the last 3.5 months, as she has been co-leading the effort that NIH has asked for to define what in fact would be the goals of the new BRAIN initiative announced by the President of the United States on April 2nd, at a time where, let's say, the general scientific ideas of what we might do with this initiative were a little in need of specificity. And Corey and her able co-chair, Bill Newsom, have been working with a dream team of really remarkable neuroscientists to put forward what we expect will be a very exciting interim plan that will guide our efforts to get this underway in FY14, which of course starts very soon. So keep your eye on the output of this deliberative process, and they will then generate by next summer a more detailed and somewhat longer-term plan for the next 5 years, or perhaps even a bit more. Because it's a very exciting time in neuroscience. The opportunity with new technologies and new approaches to be able to understand how circuits work in real time. And there's nobody better to describe how that's already coming true in an organism that we know a lot about, namely C. elegans, than today's speaker. Corey Bargmann's title is Neuromodulatory Circuits and Motivated Behavior. It's a personal pleasure to have her here today, and let me ask all of you to please welcome Dr. Corey Bargmann.
So I'd like to thank my colleagues here at the NIH, particularly Nora and Story, for this invitation. Thank Francis for that lovely introduction, and just thank the NIH in general for its support of science and, at this point, of neuroscience in ways that have just excited the whole field. I'm going to talk today about trying to break a problem down to its simplest elements. And the problem I'm going to start with is going to be complicated enough that perhaps breaking it down to simple elements is merited. And that is the biological basis of behavior. So when we look at animals exploring their environments, responding to different stimuli, recognizing each other, learning, exploring, what strikes us about them is the diversity of their different behaviors. And when we look at human beings, we also see a great diversity of different behaviors. But Going back to the 1930s, the classical neuroethologists recognized that behavior has common themes, even when its outward manifestations seem to be very different. And these biologically trained psychologists sort of very carefully examined the behaviors of different animals and were able to show that in particular behaviors associated with key functions like feeding and reproduction tended to have elements that you could recognize not only among all members of a species, but between members of different species. So for example, if you were to look at courtship behaviors or aggressive behaviors related to courtship in different animals, the actual manifestations might be very different in the behaviors themselves, but there were underlying rules that regulated when they were expressed, how they were expressed, when they would be broken off, and what the appropriate targets were. Likewise, the animal's ability to raise their offspring might appear very different in different kinds of animals, but from animals ranging from simple invertebrates all the way to ourselves, the problem of caring for the young is something that has a biological basis and certain kinds of fundamental principles that can be seen underlying it. Now, the neuroethologists had no idea of what modern biology was and how that might relate to a mechanistic basis. But this concept of ideas or themes in behavior, in particular for these kinds of instinctive behaviors, can be expressed at a molecular level. And I'd like to just mention one example of that at the beginning of my talk. And that is the small molecule neuropeptides, oxytocin and vasopressin, 9 amino acid peptides closely related to each other. The first peptides, neuropeptides actually, to be purified and synthesized from mammals, where they were recognized early on to have important roles in reproductive behavior. So when a female mammal gives birth, her hypothalamus releases oxytocin into the bloodstream. It causes the uterus to contract, driving the birth of the offspring. It also causes the milk letdown response, which allows nursing to take place. In addition, oxytocin is released into the female mammal's brain, and the release of this peptide within brain circuits provides a strong stimulus to the formation of maternal behaviors and mother-pup bonding. So oxytocin is really the one-stop shopping of mammalian behavior and mammalian birth. It gets the offspring born, it gets the offspring fed, and it gets the offspring taken care of. And vasopressin, which has more complex roles, also has roles particularly in mating and aggression related to mating in a variety of mammalian species. So a couple of years ago, a graduate student in my lab, Evan McCosco, noticed that the genome of the nematode worm C. elegans, which we study, had within it a molecule that looked to us sort of like oxytocin. Now I have to say, you had to be our very good friend to believe that this molecule was really oxytocin. It didn't look that much like oxytocin, but we were kind of encouraged because there were two potential receptors in the worm genome that looked like oxytocin and vasopressin receptors, and these really were clearly homologs of each other across species. So following on that observation, Jennifer Garrison, a postdoc who joined the lab, proved that this peptide is really made, purified it from worms, and showed that it activates these two receptors in heterologous cell systems, indicating that there really is a conserved peptide and conserved receptors that belong to this particular family of G-protein coupled neuropeptide receptor systems. So this is sort of a biochemical relationship, but what's really interesting is the biological relationship. We obtain mutations in this molecule, which is called Nimatosun, nematode oxytocin, and in its receptors. And Jennifer examined them for a wide variety of different responses. And these animals were viable, and they were superficially healthy. They were able to solve a lot of tasks, but there was one task that they really did not solve very well. And that is that the males, which are these skinny little guys here, were very ineffective when placed in a situation where they had a mating partner, the larger hermaphrodite here, and were asked to carry out a mating routine. So interestingly, they did not fail completely. They failed about half the time, regardless of whether the peptide was missing or its receptors were missing. And consistent with this function, when you look at the male tail, which has specialized neurons and circuits required for mating behaviors, both nematocin and its receptors were expressed in different subsets of neurons. So what's interesting about this from the point of view of the biology is that having a system like this enabled Jennifer to look very carefully and ask, what is the logic by which this reproductive neuropeptide is regulating reproductive behaviors? Is it required for the neurons to function at all? Is it required for mate recognition? Is it required for some particular step of the process? And so by using quantitative assays, which measured many different aspects of mating, and also by measuring things like overall mating success and the number of progeny, and even by measuring a completely different set of behaviors related to reproduction, which are food-seeking behaviors that manifest themselves over time periods of many hours, Jen found that every aspect of reproductive or mating behavior in males was present but reduced in animals lacking this neuropeptide. They seemed to act as global regulators that coordinated mating behaviors on short time scales and on long time scales of particular motor actions and even of sperm transfer. So instead of being at one level, this neuropeptide is one little element of mating behavior, which includes many neurons and many signals. And at another level, it's an element of all aspects of mating behavior that we could evaluate. So the take-home lesson of this is that it tells us something about a relationship between a molecule and a behavior that we can examine at very high resolution quantitatively. But the real take-home lesson that strikes us about this is that it suggests something sort of remarkable, which is that these molecules related to oxytocin and vasopressin may actually have been locked into a particular behavioral function very early in animal evolution, because work from a variety of people in mammalian systems, can't be here at NIH without mentioning Tom Insel's work on voles, has shown that oxytocin and vasopressin are very important regulators of mammalian reproductive behaviors, including mating. Work from Bill Kristen's lab at UCSD has shown that an oxytocin-related peptide is involved in the mating routines of annelid leeches. And our work indicates that it's involved in the mating behaviors of ecdysoan nematodes. So across all three major branches of animals, the same molecular pathways are used as global regulators of the same kinds of behavior. Despite the very different mating behaviors and appearances and mating cues of these different animals, part of the inner motivational mechanisms represented by a particular neuropeptide are linked to these systems. And we can recognize their action across a distance. And this really presents, for me, the reason and the justification for studying the nematode worm C. elegans as a model system for understanding the brain. Like all neuroscientists, I would really like to understand the human brain, particularly my husband's. And the human brain is immeasurably complex. It consists of billions of neurons connected by trillions of synapses. People say sometimes it's the most complicated object that we know of. But kind of remarkably, when we get down to the level of genes, which is what I've been talking about, there are only roughly 25, 30,000 of them, quite a small number of components that build this immense structure. And I just want to mention that humans have about 100 different genes that include neuropeptides, including, of course, oxytocin and vasopressin. So here we look at the nematode C. elegans. Now, the nervous system of this animal consists of just 302 neurons if you're a hermaphrodite, or 389 if you're a male, with about 7,000 connections between them. So anatomically, this is just a much simpler place to go. But if we look at the levels of the genetic circuitry, it's almost as complex in the number of genes. It's got about the same number of neuropeptide genes, some of which, not all of which, have pretty clear orthologs across these different species, across this great distance of evolution. And at least this part of the puzzle, we should be able to understand in C. elegans in the context of having an animal where we have identified neurons that we can recognize in every worm, known connectivity, because we know the synaptic connections between these neurons from work of John White and his colleagues, and very powerful genetic tools combined with a 3-day life cycle. So here's another illustration of that point. This is the complete wiring diagram showing the major classes of neurons in C. elegans, categorized according to their connections with each other, indicated by these edges between different nodes. And we can see that there's a structure here, different neurons connected in particular patterns, groups of neurons that tend to be interconnected. This is what we have to understand how nematode behavior works. This is what we have to understand how human behavior works. It's tremendously more complex, and these are just the large connections that you might see using a method like diffusion tensor imaging compared to the actual individually anatomically defined synapses that we can look at. So as a provisional in-between step, we think that this will help us along the path to this. So what I want to talk about today in particular are a class of molecules that represent one of the ways that nervous systems have of communicating, and those are neuropeptides and neuromodulators. So the classical tools of neurophysiology and neuroanatomy really have focused on one of the ways that nerve cells communicate, which is through fast chemical transmission, for example, with glutamate or GABA or acetylcholine at synapses. There's a second mode of communication, used a little less, of using electrical synapses and gap junctions. Superimposed on these are small molecules like dopamine or norepinephrine or serotonin, as well as these varieties of neuropeptides. Now, these molecules are often co-released with classical transmitters, but they're less accessible to the powerful tools of anatomy and physiology. They're not always released exactly at synapses, and so it's not so straightforward to determine what the targets are without testing directly. They activate G protein-coupled receptors for the most part, and that means that it's not so easy to measure directly in the form of, for example, an action potential, what these molecules have done to the target neurons within the circuit. However, it's clear that they have important biological functions, and those include the regulation of behaviors such as sleep and waking, hunger, satiety, pain pathways, bonding and affiliated behavior and reproduction, mood regulation and addiction, all of which have been associated with molecules from within this family. So their role in behavior, indicated either pharmacologically or genetically through these kinds of mechanisms, including human behavior, is very firm. So here's the question that we would like to be able to address at a microscopic level. How do these circuits interact with fast chemical circuits? What's the structure of these circuits? What distances do they act over? What kinds of time scales? How can we put together these different ways of viewing one nervous system? And I'm going to talk about those behaviors in the context of a particular kind of behavior, which is foraging behavior, exploratory behavior. And it's built on, actually, a classical observation from neuroethology, which is that when you look at foraging animals, whether those are Drosophila flying around in their cage, or mice motoring around in their home cages, or here in this case, wild terns foraging on different islands, or toddlers in a playground, you see the same kinds of patterns of behavior. You see behavior that is structured into bouts of high activity and bouts of low activity. Now, I'm showing you here circadian rhythm to show you that behavior can be structured on very long time scales, the entire 24-hour cycle. But even within an interval of the cycle, you see that there's bouts of high activity shown by these little hatch marks and bouts of quieter periods. And these kinds of activity states are sometimes called active states or exploratory states, or versus quiet resting or quiet wakefulness, for example. So we see this across a variety of different animal species. It seems to be a way that behavior is organized on relatively long time scales of minutes or hours. And Steve Lavelle, a postdoc in my lab, decided to address that in the context of a behavior that had been described previously by Manabe Fujiwara in Steve McIntyre's lab and Juliette Benarus with Didier Châtenay. And that's a behavior that has the same kind of punctuated structure, and in C. elegans, it's called roaming and dwelling behavior. And it represents behaviors that animals show on food while they're feeding. And you can see it here by looking at the tracks a single animal left in a 1-hour period, here color-coded, depending on what it was doing and how long it was doing. And what you can see is that for the first 40 minutes of this track, shown here in red, this animal moved around in a small area, not moving very far, turning very rapidly, staying in that area. But then for other intervals, like this 10-minute blue interval here, it would tear around on the food, exploring the food very widely before returning, actually, to spend another little time at this home resting place. So we can categorize these two behaviors. The animals are clearly moving during both behaviors, they're feeding, they're laying eggs, they're responsive to sensory cues, so they're not sleeping. These are both waking behaviors. We can't separate them out very easily. Animals that are dwelling are moving at a low speed and they're turning very frequently as they eat on the food, graze on the food, whereas animals that are roaming are moving at a much higher speed and turning infrequently, which is what allows them to scatter so far. So this is what that looks like. Here's a movie. There's going to be a worm right around here, if things go well, that you'll be able to see. There it is. Starting in a dwelling state, staying in one area for a while, and then roam. It just roamed. Okay. Now it's dwelling a little more. Roam. Okay. Dwell, dwell, roam, dwell. Okay. It turns out that speeding up worm movies makes them much more exciting. But what you can see is that these individual animals are sort of moving between these states, which you can actually quite readily recognize by eye using this kind of an approach. And more importantly, you can recognize it through an analytical approach to just measuring these two basic parameters, the animal's angular speed, how often it changes direction, and its locomotion speed, how quickly it's moving. And it's quite clear when you superimpose these on top of each other that there are behavioral states where speed is high and there are few turns, and behavioral states where speed is low and there are many, many turns. So these states can be categorized using a hidden Markov model, which basically says that you can categorize these states in a fairly straightforward way that the computer determines on its own into behavioral states, so that you aren't trying to figure out exactly whether one particular reversal was significant or not, and it will classify these across time. So just a couple more things to say about this. This is sometimes called in the foraging litter the exploration versus exploitation axis, which is kind of fun. And this is a behavior that reflects something about the animal's internal state as well as external cues. So if animals are in good conditions, they spend most of their time dwelling. But when food quality is low, or whether food is limiting, or if there are environmental irritants, the animals spend much more of their time roaming. So dwelling seems to be a marker of basically a good situation for the worm, and roaming more of an exploratory activity to see if there might be some better situation. So Steve set out to just develop rigorous ways of analyzing these behaviors, classifying them, and asking what the structure of these behaviors was. And what he did was to videotape many hundreds of hours of individual animals moving through these environments, and then just ask things, for example, about the structures of these two behaviors. And what he found was that dwelling states fit a single exponential with an average dwell time of about 8 minutes. Roaming states actually have a distribution suggesting that they fit 2 exponentials. There's a class of short roaming states that are about a minute long, and those are actually about 80% of the total. And then a class of roaming states that are much longer, about 9 minutes long, and those are about 15% of the total. So the significance of this is just that it tells us that we can describe these behaviors, that they're reliable, that we can fit them to a model and see if they've changed. And also that these are not, for example, rhythmic or cyclic behaviors. They don't have a regular structure like a circadian rhythm. Exponential structures suggest that they can start and end at random. So we're going to have to take that into account when we think about how they're generated. And I'm now going to tell you about 2 of the molecules that we think are important in regulating these behavioral transitions in states. The first molecule I'm going to talk about is a classical small molecule neuromodulator, serotonin, well known for its role in many biological processes, an ancient neurotransmitter shared by all animals. And I'm going to tell you from our studies of serotonin how we could see dynamic changes in particular circuit activities that underlie this behavior. And then much more briefly, at the second part of my talk, I'll talk about a neuropeptide, the 18 amino acid neuropeptide called pigment dispersing factor, or PDF. And I'll sort of relate these 2 circuits together to understand their switching and relationships to each other. Okay, so how did we get here? Steve, having described the behavior, decided to systematically examine mutants that he thought might have interesting roles in the circuitry of the behavior, which in our case involved taking classes of mutants that affected neurotransmitters, neurotransmitter receptors, G-protein coupled receptors, gap junction subunits, and other molecules that have regulatory functions in the nervous system. From this, he identified 2 molecules that had very strong effects on the duration, in lengthening the duration of dwell states. And those 2 related molecules are a serotonin receptor, the serotonin-gated chloride channel MOD1, and TPH1, the major biosynthetic enzyme for serotonin synthesis. The mutant phenotype of both of these mutants was similar, and that was, shown here for one of them, that instead of spending about 80% of their time dwelling and 20% roaming under particular conditions, these guys only spent about 40% of their time dwelling and 60% of their time roaming. Now, Steve broke that down looking at the 2 states independently, and what he found was that dwell durations were shorter and roam durations were longer. But when he looked within these roam and dwell states, their structures were normal. There were normal patterns of turns and speeds. So the microstructures of these behaviors on a second-to-second level was indistinguishable from that of wild-type animals. But the larger structure on these behaviors changed, for example, from an average length of about 2 minutes in the wild-type for roaming behavior to an average of more like 6 or 7 minutes. So it seemed to be something that was acting on a longer duration. Now here's where working on C. elegans is really helpful. We know that serotonin is an important molecule, and in C. elegans we know that it is expressed in exactly 11 neurons in 6 classes. These include neurons in the pharynx, the feeding structure, sensory neurons, interneurons, and motor neurons. So from previous work actually done by my postdoc advisor, Bob Horvitz, and in part by Rajesh Ranganathan, we know a fair amount about serotonin functions in C. elegans. It appears to mimic or imitate several responses to food. It induces egg laying and locomotion behaviors and feeding behaviors that animals show on food. And that sort of fits what we see here. We see it changing the response on food, sort of strengthening the response to good food, the dwelling behavior. So we might put an asterisk here. So we might say mimics responses to good food sources, or is part of responses to good food sources. And it has multiple receptors. We looked at all of them, multiple G-protein coupled receptors that are important. For other behaviors were not important for roaming and dwelling, but this MOD1 receptor was. So since we have only these 6 classes of neurons to think about, Steve could next ask, what's the circuit here? Where does serotonin have to be made for the animal to show normal roaming and dwelling? And he did that using the same kinds of methods you would use in a mouse to a first approximation. He had the biosynthetic enzyme for serotonin, TPH1, flanked by lock sites that allowed recombination, so that if he expressed Cre transgenes in particular cell types, he would delete the expression of serotonin in those cell types. And using that, he asked which of these cells were important. This bar graph here just represents sort of a simpler expression of the roaming and dwelling behavior. So here, wild-type animals roam about, in this case, 60% of the time, let's call it, serotonin mutants 80% of the time. And then if he inactivated the serotonin biosynthetic enzymes in either of 2 different neurons called NSM and HSN, either one caused the animal to be converted into a mutant behavior, whereas several of the other neurons were not important. Pretty much the same experiment, slightly different strategy. Look at the serotonin receptor. Where is that receptor acting to regulate the same behaviors? So 3 different neurons emerged from this study, again using Cre-Lox recombination as being important for roaming and dwelling regulation, each of which contributed to this combined behavior. So we can assemble those together into this circuit here. We can say serotonin for 2 different neurons is important in regulating roaming and dwelling behavior. And it acts on at least 3 different target neurons through the MOD1 receptor. And together, this leads to this, the activity of this circuit modifies the length of both roaming and dwelling states. So just looking at this circuit, we can already tell something that's pretty interesting to someone who spent a long time looking at the C. elegans wiring diagram. which is that this little circuit doesn't respect that wiring diagram at all. Okay, so not only are these neurons not connected by classical synapses in the wiring diagram, this violates a feature of the wiring diagram that's been commented on by everyone who's ever seen it, which is that it is strongly feed-forward, going from sensory neurons to interneurons and integrating neurons to motor neurons. But in this case, we have a motor neuron in the pharynx, although it may also have some sensory roles, and a motor neuron in the body that are releasing serotonin, and they're feeding back on interneurons and even on sensory neurons. So they're running in reverse compared to the overall structure of the worm's wiring diagram. So this is different from the classical wiring. Next question. What does one molecule like this do in real time? Can we relate the activity of these neurons and circuits to what the animal is doing? So to do that, Steve imaged calcium in the foraging animals as a surrogate for neural activity by expressing the GCAMP calcium indicator specifically in the NSM neurons, one of the serotonin-producing neurons in the head of the worm. And he did this with an imaging system developed by Dirk Albrecht and Johannes Larsch in the lab that lets us simultaneously monitor calcium activity and the animal's movement. So that when we look at that little neuron in the head here, we can watch the worm moving around. We can see a little change in reorientation, in this case actually associated with a little bit of activity of that neuron. We can track the animal here. We can track the fluorescence and therefore calcium activity of the underlying neuron, and we can relate those together. And so Steve used these methods to image NSM calcium, the serotonin source, in foraging animals. And he saw that there were sort of events where fluorescence increased, and there were events where different behaviors occurred, and there was obviously some complexity to the structure. So he took 100 traces of this sort where he tracked animals for 30 minutes or an hour, and he aligned them to each other using two different criteria. He said, look, I see a set of events in which NSM seems to have calcium peaks. And so he aligned all of the traces based on when a calcium peak was present. And then passively, he asked what happened to speed in sort of an event-triggered averaging approach. And what he saw was that these calcium peaks were associated with a drop in locomotion speed that recovered on a similar time frame. And note that these things are slow. They are about 2 minutes long, which is, C. elegans neurons are slow, which means maybe they operate on the time scale of a second. This is very slow, even for a C. elegans neuron. And conversely, if he aligned all of the traces based on when animals started roaming, which means they were moving quickly, he saw that calcium decreased in the NSM neuron before a roam and then recovered as the roam ended. Is this related to serotonin, or is this just a coincidence? To get at that, Steve looked in the serotonin mutants using the same approach, and what he found is that NSM activity was a much poorer prediction of roaming behavior in these mutants compared to the wild type. So for example, there still were calcium pulses, but not nearly as well synchronized as speed decrease. There were still some roaming events, but now you no longer saw the dip in calcium before roaming occurred. So serotonin does seem to have a causative role in the behavior, aligned on these timescales. And finally, Steve used channel rhodopsin, expressing the optogenetic tool within the NSM neurons to artificially stimulate the release of serotonin from those neurons, or he used optogenetics to mimic the activity of serotonin on the target neurons. This is an inhibitory connection. It's a chloride channel. So the tool that he used here was ARCH, an inhibitory transmitter to imitate what serotonin, inhibitory channel to imitate what serotonin would do. And in either case, he saw the same result, which is if he took a population of animals, which he aligned for those that were roaming when he turned the light on, and then asked what happened, turning the light on led to a rapid transition of many of the animals into a dwelling state compared to control animals, indicating that activating these neurons or inactivating these neurons was sufficient to drive dwelling. And interestingly, the dwelling state continued for about a minute after the light came back off. So the dwelling state outlasted the stimulus that triggered it. And this next experiment is another optogenetic experiment. It's actually my favorite result of these experiments. And in this case, what Steve did was he took animals that were already dwelling, and he activated
the serotonin system with channelrhodopsin, or he inhibited the target neurons, and he asked what that did to the structure of the behavior. And what he found is that if animals were dwelling and you activated these neurons or inhibited these, their next dwelling state was greatly prolonged, that 1 minute of light led to a dwelling state that was 6 minutes longer on average than those that were seen in the absence of light. So it really seems to not only initiate dwelling states, but prolong them if they're pre-existing. So this is really the conclusion of this part of the talk. We can say that this neuromodulatory transmitter induces and extends dwelling states. These are long-lasting behavioral states. They last about 8 minutes long. And serotonin is capable of at least at the level of prolonging dwelling states on a similar time scale of about 6 minutes or so. So the other behaviors, the behaviors associated with dwelling, the kind of quick motor actions, are taking place on a time scale of seconds. This much slower effect is being mediated through dynamic changes in the modulatory state. Now, there are a couple of other things to say about this. I mentioned that roaming and dwelling were regulated by things like nutritional cues and various sensory cues. Many of the neurons in this circuit are known to detect subsets of these different kinds of external environmental cues. So it's possible that this circuit represents a place that information about the environment is converging with information about modulatory state to regulate behavior. But none of that has been tested yet. Okay, now on to the second transmitter, the pigment dispersing factor, or PDF. So PDF was first identified in crustaceans and in Drosophila. And one of the results that we think is really resonant with what we're studying here is a result initially from Paul Taggart's lab, in which he noticed that Drosophila PDF seems to be important in coupling circadian rhythms to activity levels. So if you look at a wild-type fly or a PDF mutant, you can actually see that PDF mutants still have circadian rhythms. They're still having bouts of high activity at certain points in the circadian cycle. But the structure of those rhythms is disrupted. So in a wild-type fly, for example, there would be a lot of activity coupled at this particular time in the circadian clock. In PDF mutants, it's sort of distributed all over here. So there seems to be something about activity levels and activity bouts and PDF that's important in Drosophila. And in C. elegans, what PDF seems to be really important for is roaming behavior. When Steve looked at mutants in the C. elegans PDF peptides or in the C. elegans PDF receptor, these animals just spent almost all of their time dwelling and almost none of their time roaming. So that represents about 95% of their time dwelling, staying in one place. Now, when the animals were roaming, their behavior was pretty much normal. It's not that they couldn't move. If you took them off food, they moved very quickly. They showed normal off-food behaviors. It just seems to be the transition here. And again, we can see that transition in the duration of the roam and dwell states on these long, minute-long time scales. That PDF receptor mutants will dwell for 15 minutes at a stretch, longer than wild-type animals. And conversely, PDF receptor mutants only show short roaming states. They'll only roam for about a minute or so, whereas wild type will often roam for several minutes at a time. So these are exactly the reciprocal effects from those that we saw in the serotonin mutants, expressed in the same set of behaviors. And the conclusions that we drew out of looking at the serotonin receptor mutants and serotonin mutants were echoed by the conclusions that Steve drew from making the same kinds of cell-specific knockouts and cell-specific rescues of PDF and the PDF receptor. He found that PDF is expressed from multiple neurons, and a subset of those neurons are important in regulating roaming behavior. And PDF receptor is actually expressed in many neurons, but again, a smallish subset of those neurons are important in regulating roaming behavior. So in each case, it's not one neuron that's important, it's not all of the neurons that are important, it's some, but that particular patterns that regulate these transitions. Steve also addressed the function of the system using optogenetic approaches. Now, PDF is a classical neuropeptide, and it acts through a classical neuropeptide receptor. It's been shown in heterologous systems that this activates the GS adenylyl cyclase pathway to lead to the production of cyclic AMP. Now, using optogenetics to depolarize or hyperpolarize neurons would not necessarily imitate the effects of cyclic AMP on those cells. So to use optogenetic interventions here, Steve took advantage of a bacterial light-activated adenylyl cyclase to perform optogenetic activation of cyclic AMP in target cells, planning to imitate the functions of PDF in those target cells. And what he showed was that if he took animals that were dwelling and he induced adenylyl cyclase in the neurons that express the PDF receptor, he saw a rapid increase in speed and decrease in turning that indicated a transition from dwelling behavior to roaming behavior. And as was seen for serotonin and the opposite behavior, the roaming behavior outlasted the stimulus that induced it, that it was evident for at least 2 minutes after the stimulus was removed. So 2 different neuromodulators, one neuropeptide, one small molecule, 2 opposite effects on behavior. How do these relate to each other? Are they in series, that one regulates the other? Are they in parallel? To address this, Steve used the technique of double mutant analysis to make animals that lacked both the serotonin receptor, MOD1, and the PDF receptor, and looked at their behaviors in roaming and dwelling assays. Now, at first glance, the behavior of these double mutants seemed sort of like wild type. They roamed about 70% of the time, they roamed about 30% of the time, and dwelled about 70% of the time. But closer inspection actually revealed something quite different. So when Steve looked at these animals in detail, he found that the double mutants had the very short dwelling states characteristic of serotonin mutants. And they had the very short roaming states characteristic of PDF mutants. So these animals have unstable behaviors. They rapidly switch back and forth between roaming and dwelling behaviors. They're unable to maintain a stable behavioral state. And so looking at this double mutant, our interpretation is as follows. The primary role of these modulatory systems is actually to extend these behavioral states. The behavioral states can exist without them. The behavioral states can even percolate along for a minute or so without them. But if you want to have a long-lasting, 8 or 10 minute long behavioral state, then you need this parallel modulatory system to maintain it. And in addition, they appear to inhibit each other in these different roles to help to set the overall level of these different states. So in conclusion, what we found here are 2 distributed circuits for foraging behavior. And we call these wireless circuits because the neurons that make serotonin are not connected by classical synapses to the neurons that respond to it, and the neurons that make PDF are not connected with classical synapses to their targets. So the classical wiring diagram does link elements within these circuits, but not in the same pattern as the modulators, and therefore wireless, not wired. Functionally, each extends one behavioral states. And I want to mention just one more thing about these. The effect of killing any of these neurons is much stronger than the effect of removing the modulator. So if we kill, for example, the ASI neuron, that's a much more serious defect than just hyperpolarizing the ASI neuron with ARG through this pathway. So these neurons are doing other things in addition to what the modulators are doing, presumably through their other effects in the classical wiring diagram. And as we look at this, here I've superimposed those circuits back on the wiring diagram of C. elegans that we started with, the classical anatomically defined wiring diagrams. And the point, so what I've done is to circle the serotonin and PDF circuits here in blue. And the point I want to make here is that you could not have chosen a more random subset of neurons from this wiring diagram in terms of their connections, their locations, their apparent functions. This is really a separate logic. The logic of the neuromodulators is a logic that will have to be solved independently of the modulators of the fast circuits that generate locomotor patterns. They do overlap here in some of these green neurons down here, for example. Ultimately, some of these targets are the same, but the connections between targets are different. And the conclusions intellectually here, for each system that we look at, multiple neurons make the modulators, multiple neurons make the receptors. Each neuron has other independent functions. So if we look at neuron and modulator and receptor as a Venn diagram, none of these are subsets of the other. Each has to be solved as a separate problem. Second, these signals are not just sort of in the bath, they're actually instructive for generating new behaviors. They induce behavioral states and they stabilize pre-existing behavioral states. And our way of thinking about what these guys are doing, particularly in the stabilizing state, is that they're converting electrical time into biochemical time. Fast circuits are able to maintain persistent states for tens of seconds. But when you're starting to get into tens of minutes, then things like G-protein pathways and protein phosphorylation are much better ways to stabilize, to hold biological information than these sorts of rapidly changing neural activity patterns. And finally, little connection with the direct connections in the wiring diagram, separate and orthogonal circuits. These are the lessons we have from C. elegans. Should you have to think about these lessons if you work in other kinds of nervous systems? Now, the roles of modulators are very clear across the nervous systems of invertebrates. And I'd just like to point out that the more we understand about the functions of vertebrate nervous systems, the more we understand the roles of modulators as being highly important. So within the vertebrate retina, dopamine, the neuromodulator, is present as a regulator of circuit states. The retina functions in very different ways during the light, when cone photoreceptors dominate the circuits, and during the dark, when rod photoreceptors dominate. They're much more sensitive, but they have less, these circuits have less spectral and spatial resolution. The switch between these mechanisms is made by dopamine acting at multiple points in this circuit to effectively switch the circuit between night vision and day vision. So these are, again, long-lasting circuit states happening in parallel to the very fast computations that can occur within the synaptic connections of the retina. And since someone I was at a talk with recently said that the retina was an honorary invertebrate, which I just thought is special pleading. In any case, I just want to point out that whatever part of the brain you're interested in, there are neuropeptides and neuromodulators that act in that part of the brain. And that includes the highest brain areas. And they're prominent in cortical circuits. I started with oxytocin, and here I'm sort of ending with oxytocin on a recent paper from Dick Chen's lab looking at the role of oxytocin in the hippocampus, where there are thought to be roles in memory formation, where he showed that oxytocin, acting on oxytocin receptors, very specifically modulates fast spiking interneurons to change spike transmission, again, to regulate the flow of information through the classical circuits. And then this slide below here is actually a recent circuit mapping slide from Massimo Scanziani's lab, in which he was looking at the roles of different inhibitory interneurons, their connections with one another, and their connections with excitatory neurons, mapped out through optogenetics. And the point I want to make here is that neuropeptides are so prominent in the cortex that we name cell classes by the neuropeptide that they express. That people call these guys here the somatostatin interneurons, and these guys here the VIP interneurons, because that's the best way to identify them. Now, it hasn't actually been characterized what these neuropeptides are doing in these neurons, which also express the neurotransmitter GABA, but their expression here is quite prominent and reproducible. And I would just like to really highlight VIP, vasoactive intestinal peptide here. The VIP neurons have an interesting place in cortical circuits. They're a place that subcortical information about arousal and cholinergic input comes in to regulate the activity of local circuits here. In addition, VIP itself and its receptor, Viper2, are recent hits that have emerged from large-scale genetic studies of human schizophrenia as risk factors predisposing a certain number of humans to alterations in their cognitive states. And so I don't think it's a good idea to ignore these molecules. I think that we have much to learn from them as they are embedded within the circuits that we have studied so powerfully for so long, but performing perhaps different functions. And with that, I will just thank you for your attention and show you the pictures of people in the lab who've worked on this project. I've always thought that the secret to my success in science has been my good taste in people, and I feel that that's really reflected by the scientists who've done this work. The work I talked about was really almost entirely the work of Steve Flavell, a postdoc in the lab, who's been exploring quantitative methods of addressing these modulatory circuits. Several of the tools he made were developed by Naveen Pokala, a postdoc in the lab who's sort of our basement tinkerer who makes lots of cool molecular tools for monitoring neural activity. Dirk Albrecht and Johannes Larsch here engineered the imaging system for doing real-time imaging of neural activity and behavior. Jennifer Garrison's work on oxytocin I talked about just at the beginning of the talk. Thank you very much.
Thanks for a very interesting and somewhat surprising talk in terms of some of the connections that you unearthed. We have time for questions. People, please use the microphones so those watching on video can hear the questions, and we can start right over here.
One of the most important sources of neuromodulators in the vertebrate nervous system are glial cells, and the morphological changes in astrocytes regulate oxytocin, and pain and sleep, etc., are regulated by glia. So I'm wondering what you think, how important you think glial cells are in modulating neural circuits, and I'm just wondering why they're not mentioned in C. elegans' work.
So we're actually, one of the things about being a geneticist is that you kind of go in agnostic to where your gene is acting. You go and you look at where it's expressed. These particular molecules were not expressed in glial cells, but if they had been, we would have followed that up. Not much has been done on C. elegans glial cells. My colleague at Rockefeller, Shai Shaham, has been the group that's done the most and has identified a variety of their functions in development, some of which are quite similar to those that are known in vertebrate systems. And I think he has also shown that they have roles in enhancing neural activity. What those are, right now don't involve these particular modulators and these particular behaviors, but I would stay tuned. Ed.
Hey, so you mentioned that the signals are going to a C. elegans person the wrong direction, from the motor neurons back up to the inner neurons and such. Does that matter? Is there a logic you can make sense of for why those are the cells that are providing the PDF or the serotonin? And is it just an anatomical logic? Those are the cells that are used because they're in the right place.
You know, I don't have anything to say. I don't think we understand the logic of where these guys are being made at this point. And I think we have to think about that more carefully because these molecules have other functions when they're expressed from other neurons in other behaviors. So PDF has been shown by Maureen Barr's lab to affect male mating behaviors. That uses a completely different neuron from source of PDF and completely different neurons expressing the receptors from the ones that Steve defined here. Serotonin affects a variety of other behaviors, again, often through sources from other neurons and receptors on different neurons. So we think that, you know, these cues are neither acting at a complete point-to-point level, nor are they acting as sort of global sprinkler systems, to use one of the old terms from neuromodulation. There is a selectivity in the way they're put together. And what that represents in terms of space and levels, I think is an important question to be worked out. So I think it will have to do with the source, how much is released, how far away the receptor is, what the efficacy is of that receptor, and that will represent some sort of a decoding of these signals. But we don't have any insight into what that is yet.
Right, so you don't know yet about how much of that regulation is in terms of the distance spread, or is it a gating as far as when the neuron is, the receiving neuron is ready to receive the signal? So it could be a temporal coding or a spatial coding. You don't have a sense of that yet?
No, we don't. We haven't done sort of comparative behaviors yet. Yeah.
So behavioral states in animals are often a mixture of both changes in activity and changes in sensory responsiveness. And so this kind of backwards flow makes a lot of sense in terms of modulating sensory responsiveness. Do you think it's possible that that actually drives then the changes in activity? Could it be that because the states themselves are driven by particular environmental signals, you're changing the sensitivity of the neurons that are sensing those environmental signals?
There's some very nice work I'm coming up with an example from David Anderson's lab, but there are results from other systems as well, showing that neuromodulators can, in fact, change the sensitivity of sensory neurons, or their ability to couple, I would say, more to downstream circuits. So what David showed is that in hungry flies, dopamine is produced, and that dopamine acts on the terminals of the sweet-sensing neurons to enhance synaptic transmission, and therefore make the flies more sensitive to sweet food when they're hungry, okay? So it's not in the periphery, but at their first synaptic input. So I think that's an excellent example. We have other examples, I think, that we don't understand as well, and I think there are examples in other systems. So yes, I think the principles of how these systems are changing will in some cases involve changes in the peripheral detection of cues, and other places changes in the dynamics of the internal circuits that are relevant to the behavior. I think we'll have to interpret those where they arise.
This is slightly related, this question, but in your optogenetic experiments where you either stimulated the serotonin neurons or inhibited the receptor neurons, have you played with the environmental conditions? So when you induce the roaming or the dwelling to roaming, if you do it in kind of tainted food, do you have a harder time making that switch?
Yeah, those are good experiments. We haven't done that. We haven't started playing with those, with the intersection between all the other forms of regulation yet. Yeah.
And then can I ask one more quick question? Sorry. But given the conflicting roles of oxytocin in mammals in its role in feeding behavior, did you see anything, when you were looking at your mating behavior and your oxytocin mutants, or when you made your rescue experiments, have you ever done constitutive active oxytocin receptors in C. elegans and seen any differences in feeding behavior?
So we have not seen differences in feeding behavior. We have actually seen differences in some thermal stress-related behaviors, which vasopressin has a role in stress-related behaviors in mammals, but that's unpublished. Liliana Schuf's lab showed a role in some learning behaviors, but again, not explicitly feeding related. So it might come around. You may have to sort of zoom in on particular elements, but that, there may be sort of pieces that are conserved and pieces that are not. Evolution has a maddening combination of sort of maintaining functions and then just sort of opportunistically making something new that tends to laugh in the face of your conceptual divisions. So this may be one of those.
Great. Thank you.
So besides roaming and dwelling, is there any other intellectual activity these guys could do?
So in terms of behavioral states, they also have a resting state, so a sleep-like state. They have different foraging behaviors off food, where they first search for food locally and then go into long-lasting dispersal states. These are these sort of long-lasting, 10-minute or more, 10-minute or longer behavioral states. The other thing that C. elegans is actually it's motivated on shorter time scales to do a variety of sensory behaviors. It detects thousands of odors, it chemotaxis to odors, it learns to avoid odors that appeared when it was starved, it learns to avoid odors that were subsequently paired with pathogenic infection. So it learns to track to a preferred temperature. So there are sets of behaviors, there are pheromone-related behaviors as well. So there's a fairly, it's a compact diversity of behavior. Nobody's going to teach C. elegans to speak French or play the piano. But in terms of having, some of the basic elements that you might think about, that you might be interested in the nervous system, just the building blocks of behavior, such as sensory behavior, sensory discrimination, learning, associative and non-associative, other forms of plasticity, multimodal integration. Those things do exist in this system. So it's a place to think about just to mix metaphors. It's a place where we can sort of think about the grammar and the syntax of behavior, sort of primitive elements, but these are not the works of Shakespeare.
So considering few number of neurons, about 300, you think, and 20,000 genes, so each of these cells have multifunctions. You think they could deal with it effectively, or everything is done at a much slower level?
The neuron, so I suspect that all of the neurons are multifunctional. I think that they're forced into that by the small nervous system. However, multifunctionality is seen in a lot of nervous systems with many, many more neurons. The little retinal subcircuit I showed there, actually, the certain elements of that circuit are shared between the rod and cone visual systems, which they wouldn't have to be. So those elements are, in fact, multifunctional elements. So I suspect that that is a feature of other nervous systems, but a very prominent feature of C. elegans nervous systems. They are slow. No one really knows why. My thinking about it is that they do a lot of they have graded potentials and do a lot of local computations. That kind of information is lost if you have spiking neurons and action potentials. So they may have sort of chosen to go for having the largest sort of diversity of computations within their neurons rather than having the fastest transmission of information.
Thank you.
Let me ask the last question. So obviously, people will argue really understand a system when you can model it and predict behavior based on what you know. And obviously, of those who would love to build a computational model of the human brain should be sobered by the complexity they're facing, and maybe ask the question, how far away are we from having an effective, detailed computational model of C. elegans that you could trust to actually predict real behavior most of the time? How are we doing?
That's a great question. Mita Chikalovsky's lab has a pretty good model for sinusoidal locomotion. And that's really the motor pathways that just let the worm move up and down like that when it moves forward. And that's about it. People are starting to think about models for little chunks, sort of like little motifs within the wiring diagram, and what those are doing. Very much along the same lines as people do in systems biology. And some of those have started to have some predictive power that you then test with experiment. You think, oh, this is right. So I think the ability to combine model and experiment is sort of moving up here. The initial models, I should say, when people just looked at the wiring diagram, people tried to model it just based on anatomy, and that didn't yield a lot of insight. You could get from everywhere to everywhere. I think the answer to that is that things are multifunctional. They operate on different time scales. They're regulated. The strengths change depending on things like these modulatory states. So it's a set of possibilities. It's not a single answer, and I think that's what led people. That's what prevented people from looking at that first step. So I think we need to impose activity on that connectivity.
Well, there will be a reception in the library, and you'll have a chance to speak in greater detail to the speaker if you would like. But please, let's thank Dr. Bargmann again for a very stimulating lecture.